For non-model organisms, there's currently a limited number of data capture methods with the goal of examining subsets of the genome for phylogenetic or phylogeographic analyses. Anchored phylogenomics and ultra-conserved elements are two commonly used genome subsets. Our goal was to find a subset of the genome where each locus produces a reliable gene tree, even for recent divergences, where the sequences are present in all squamates, and the sequences can easily be aligned and easily partitioned. To achieve this goal, we used two basic criteria. First, we chose protein-coding exons 1,500 bases or longer. Second, the exons had to be more variable than RAG1 based on pairwise comparisons of DNA distances. We call this approach RELIC, which coincidentally is a popular mosquito repellent in Europe. I don't know what that means, but it's true. Our subset includes 219 exons spanning 703,000 bases. The average exon size is 3,300 bases. Many of these exons were not easily found because the anolis genome and most of the squamate genomes annotated using anolis are incorrectly annotated, especially in the case of long variable exons. This is one example, EXPH5. Over half of all our loci were incorrectly annotated, but were confirmed to be single long exons using transcriptomes. As a proof of principle, we wanted to compare RELIC to anchored phylogenomics and ultra-conserved elements. Using R scripts that iteratively probed 12 complete squamate genomes, we restricted our analyses to loci that could be found in all 12 complete genomes. We compared our set of 219 exons to 1,642 UCE loci and 406 anchored phylogenomics loci. We used 10 publicly available genomes and 2 unpublished gecko genomes and transcriptomes provided by Tony Gamble. Our comparisons included concatenated species tree and individual locus informativeness. Perhaps the clearest difference between our exon set and our other genomic subsets can be seen when examining phylogenetic informativeness using Townsend's criteria and using the time-calibrated tree shown here. The x-axis is time, and the y-axis is informativeness, and each colored line represents one locus. I was a big critic of this approach until I saw these results. Pause for laughter. Within all three subsets, concatenated and species tree topologies were identical to each other. However, the anchored phylogenomics data produced a different topology than UCE and RELIC, whose topology is represented by the time-calibrated tree shown. The difference in topology for anchored phylogenomics was the relationship of dopasia, and this inset shows the alternative anchored topology. Here's a comparison of species tree results shown above the branches with the per locus support for each branch shown below the branches. This is nearly the same as the majority rule consensus, but not quite. In every case, RELIC has the highest per locus support. Particularly note the most recent divergences, which is what RELIC was intended to resolve. The sister taxon relationship of Pantherophis and Thamnophis is supported by 87% of the long exons, but less than 70% of the other two subsets. In conclusion, rapidly evolving long exons are a new subset of the genome that produces highly resolved individual gene trees when compared to other popular capture methods. RELIC can be easily partitioned and further analyzed using codon modeling. Here you can see that two loci, BRCA1 and BRCA2, have great potential for reliable gene trees because all three codons are changing at about the same high rate in contrast to nearly every other commonly used marker that's exemplified by RAG1. The third codon position makes up most of the information. The general approach of seeking long variable exons can be applied to any group that has at least two genomes for references. I'm currently working on a set for frogs. Our goal was not to resolve squamate relationships, but to find a set of markers that will be useful at many different levels, all the way to closely related species complexes. Special thanks to Margarita Metalanu, Tony Gamble, Shannon Keating, NSF, and Villanova University.